Great. So joining me for the next hour or so is Jo Verran, who is affiliated with Manchester Metropolitan University and the FEMS Education Group, but also IBBS, which is the International Biodeterioration and Biodegradation Society. We've already had two of your members speaking.
Yes, that's right. Yes, our society's been very active in the International Microorganism.
And actually fascinating, because it's not a topic I know anything about particularly, but when you delve into, you know, because it's so much about our built world and our human world and how it's being changed by microbes, it's fascinating.
It is. It's a really nice area of applied microbiology, yes.
So yeah, you're here to join me today because we have, well, let's frame this a little bit. We want to kind of argue the case that we want to be using FEMS as a hub in some way for educational resources. And we have several different resources that we've curated and collected, which we're going to go through. But you also have other initiatives that do a similar role, like the Bad Books Club. So I wanted to know if you wanted to say a word about the Bad Books Club and your activities today before we set off on our educational adventure.
Yes, I will. I also would like to say before we start that I think there are many microbiology academics who have loads and loads of fantastic ideas and have done lots of different educational activities. And I really encourage them as much as possible to tell other people about them, because the breadth and the scope of activities that I know about is really significant. So talking about them today, some of them, and also encouraging people to think about disseminating them more widely is, I think, a great point for today as well.
Absolutely.
First. So I've done quite a lot of educational activities in my career. And when I was teaching also, I tried to encourage my students to think about how they could communicate their science as well as kind of learn about it. So I encouraged them to use art as a way of exploring science or explaining science to others, microbiology particularly. And from there, I kind of went on to look at literature, and I wanted to look at fiction rather than non-fiction literature, because I felt that fiction was something that anyone who enjoyed reading could engage with, so they didn't necessarily have to be scientists. So it's called the Bad Bugs Book Club, and I set it up in 2009. So it's been going for 11 years now, so it's can't have been doing everything wrong.
There are human beings that can have conversations who are younger than your Bad Books Club, so that's quite a good achievement, I think.
Yeah, that's good. And we have had some books for younger people as well as part of the Bad Bugs Book Club activities. So my aim originally was to get scientists and non-scientists together talking about fiction novels where microbiology or infectious disease formed part of the plot. And I have a website, the Bad Bugs Book Club website, and on that website, for every book that we have discussed, I post a reading guide, so there's questions that people can use in their own discussions, and also meeting reports. So the website is there as a resource, really, for anyone who wants to set up their own Bad Bugs Book Club. And the Microbiology Society actually this year is also looking at the Microbio Book Club too. So just trying to encourage people to explore fiction as a way of talking about infectious disease, I suppose, in fact, really. So we've read more than 60 different books over these years. And, and this evening we're talking about The Constant Gardener by John le Carré. So we've got a small book club meeting, but then at 6:30 UK time, but then at 8 o'clock, I'm just going to post a few questions on Twitter so people can join in if they want to, or just see what the questions are and the discussions. Visit the website.
Hopefully they can watch the film and get involved.
Yes, that's right. Yes, yeah.
I have seen the film a long time ago, but maybe I'll watch it again tonight when I'm finished with this live stream.
And join the Twitter.
Yeah. So speaking of educational resources, I mean, so one of the things that we want to highlight and kind of, you know, help provide is that we have an education section of the FEMS website, which is slowly growing with resources. So we need to improve its visibility, but for the meantime, you can get there by going to the opportunity section, going to our resource board and clicking on education. And on here, there's several amazing initiatives which I want to sort of show, and then hopefully we can inspire more people to add their resources to the board. And I'm sure, like you say, there are so many academics who put time into these projects, and we really want to share them with as many people as possible. So the one I'm going to start with is one that for me is personally the most exciting, or not the most exciting, but at least an exciting one, because it's one that we worked on here in FEMS. So last year, over the past 12 months, we made videos around microorganisms, the 52 Microorganism Project. We posted them once a week on social media, and they were shared far and wide. This is quite topical in the sense that the most well-watched video was Magnetospirillum, a magnetic microorganism, which we just heard about with Pedro. But now what we've done is we've taken all these videos and we've put them as a resource on the website. And you can now come here to the open source project and you can click on this link and you can download any of the videos that you want to share with people. So we have all the different microorganisms, 52 in total. And these are really great to share on smartphones, and they're quite useful for children who might want a more digital way to interact with microbes. And we've created 2 games. So you can do these with a class or with a student body. And the first activity is competitive trading. So if you give these video files to your children, 1 to each student, and they have to then trade the files to try and get the full collection. And then they must come to you if they have all 52, explain their favorite one, and you can give them a prize. And you can set a time limit, or perhaps even ask them to present on the microorganism they found the best. And that's the second activity, is collaborative presentations. So you can provide all the videos to a group and then ask them to look through them for their favorite one. And then they have to present about them, but they also have to present information that isn't in the video is the crucial thing. So they have to do some research. So this is all available on our website. You can download any of the videos and yeah, try and share them far and wide. This is a FEMS initiative. Oh, sorry, go on.
I was going to say it's a really nice initiative, and I like particularly the ones that for some of the larger microorganisms, there's some really nice moving images. But I also like the music, and I did notice that there are different tunes. And did you think the tunes, or is that just something?
The tunes we got from, actually, I've just got one in the background here. The tunes we got from just a stock image website. But I tried to pick ones that were a little bit more fun. But this one here is this is the most well-watched video. This is on the magnetic microbes. So I'm now surrounded by them. And we heard so much more about these from Pedro earlier, but this gives you a brief introduction to the topic. And they're all less than a minute long. And so they're very easy for people to share on social media and smartphones. And the idea is that, by responding to new technology, we doesn't mean that it's more difficult to educate people in any way. It actually can be faster and more fun.
I also really like the diversity of organisms that you chose. I think you don't just pick the pathogens and everything.
No,
you have a whole diversity of different groups and different applications, and really unusual ones like this one. I think it's a really nice idea.
It was great fun to make them, and we I'll just stop this playing on loop because it will get distracting. But we crowdsourced a list of microorganisms from staff members and their families.
Okay.
So I think that helped explain the diversity of microorganisms.
Yeah.
But there'll be many people's favorites in here somewhere. So if you're a teacher or you're just interested in learning about microorganisms, this is here on our resource section.
Yeah, and I also, another activity you could get students to do is to think about, have they got a favorite microorganism that's not there, and, create their own.
So this is one of the things we've done. So actually, and it will require a little bit more development before it's exactly what it intends to be. But within the same open source folder, we have the editing and asset files. So if you go into this folder, you can download the artboards and the Premiere Pro project file and a few other assets to perhaps start editing your own videos together. And then this is going to be developed further, but the idea is that we can get million microbes. So we can go beyond 52 and try and get millions of different videos in the long-term future. Yeah. So you can download the asset file.
Anyway, I suppose, isn't there?
Exactly. You've got to aim high. And then have a go editing your own videos if you have Adobe software at hand, which you can get usually from a university or a school. And
so students could do stories about the germs or the bugs that they're doing their projects on, their final year projects, or their, or school projects or PhD projects. So
well, anything you're working on in the lab.
Yeah.
So this is just a brief overview of a FEMS Education Initiative, but I have several others by other people on this website. which I also want to look at. And the next one is one of my favorites in a huge way, because, let me just stop this sharing because I need to open up a new thing. It kind of gives, we're talking about microorganisms all day, but one of the really important things is to try and understand, well, how tiny are these organisms? And this is what the next resource tries to do, is to show everybody the true scale of the universe. So it's called the Scale of the Universe 2. I guess there are a couple of versions. It's created by Carrie Huang and implemented by Matthew Mattori. And yeah, it's a wonderful, wonderful resource. And let's see if it loads up here, because it's quite fun to share. So we start at our normal human scale. So, you know, human-sized things. But this allows us to zoom in to an unprecedented microscopic world, and kind of put these things in perspective. So we're starting here at a position where, you know, 1 meter is a perfectly normal length. Giant earthworm, I didn't really realize how big they are. Also being here, shown for scale, dodo, beach ball, the Raphcelia, which is the biggest flower in the world. But we can zoom in and start to see things at different scales. So is this coming through for you okay, Jo?
Yes, it is, yeah.
So, you know, small but still very normal things here. Eggs, earthworms, matchsticks, pennies, square inch, coffee beans, grain of sand, ants, duckweed. This is probably where we start to think about things getting towards the scale of too small to see. But beyond this point, we really do get to microscopic things. And now at the scale of a millimeter. So we have, you know, some familiar things, grains of salt, grains of sand. Perhaps you can stare at your screen and find an LCD pixel. But then we also have the largest bacteria and amoebas. And what I find amazing is an amoeba and a bacteria are single-celled organisms, as we like to know, but they're the same size as dust mites, which are multicellular, which kind of blows my mind, because they must have huge cells in comparison to these dust mites. And these single-celled organisms are on the same scale, and that's just a bit weird for my mind. But as we keep going, we can see smaller and smaller things. Human eggs, width of the human hair, silk fibers, skin cells, and mist droplets. And now we're starting to get to really microscopic things like white blood cells, chloroplasts, even organelles within the cells like mitochondria and cell nuclei. But we can keep going further. So this is E. coli, our favorite, well, some people's favorite model microorganism. But what's interesting is it's about the same size as X and Y chromosomes. So the one molecule of DNA in your cell, when it's all compacted up, ready for mitosis, is about the same size as E. coli. So E. coli really are tiny. They're so much smaller than our normal mammalian cells. Not that we're normal. Maybe they're normal and we're the weird ones. They are very,
more
very
than us, so don't argue.
But we can keep going, and we can see the largest known viruses, a little bit smaller than E. coli. Obviously, viruses very present in the news these days, but these are other kinds of viruses which are much larger. And we can also then see that there are bacteriophages, which are viruses that work on bacteria. And we're smaller than single-celled organisms here and at the kind of border of life. So at this point, we're starting to get a bit confused as to whether these things really are alive. Because they get so small, they just have so few atoms that it's hard for them to do all the things that we would classify as life. Beyond this point, we start to get out of the realm of microbiology and into physics, but it's still no less interesting. This is a transistor, which you might find in your smartphone. And then we have, a few macromolecules, which are obviously very important to life. DNA, phospholipid bilayer. And we can keep zooming in, and then we find the individual macromolecules. So you might have glucose, important sugar for keeping life going, lovely, delicious glucose, provides lots of energy, and a single phospholipid molecule that sits in the membrane of any cell. And then we can keep going even further, and we get into the scale of atoms and subatomic particles. But I think this is a really useful tool for showing the scale of all these things.
I do, and it's always very difficult, for example, some of the public engagement activities we do to kind of conceive of the size of a virus particle, and we do all sorts of different activities to try to say, well, you know, this is a human cell looked down a microscope, and now we've made a giant poster of a human cell, and this is a virus in that cell, and you can make a virus out of plasticine and that sort of size. And then, and so It is difficult to conceive of, particularly the size of sub-microscopic particles, I think. So this sort of visual is really interesting, really nice.
Yeah. And I do encourage anyone to play with it or share it with your class or friends, because it's just, one of these, once you have this in your mind, then it never goes away in a sense.
No, it's really useful, yeah.
Yeah, and we'll keep zooming out just to kind of put everything into perspective again. But yeah, as we get back towards the more normal method, I'll just mention that this does go the opposite direction. So we can also zoom out and start seeing bigger things. And this gives you the sense of scale in the opposite direction. Going on to human objects, the largest things we've built, pyramids, Eiffel Towers, that kind of thing, Hoover Dam. And then it starts to show the scale of asteroids, countries, mountains, this kind of thing, small planets. And then we go out further, we get kind of bigger planets and moons, perhaps. There's the Earth for scale, smallest stars, biggest planets, and then we start to go into stars, and we keep going out, and we can eventually reach even huger objects like, nebula, light years. Getting out of the realm of microbiology here, but it's still pretty cool. And then we can go out all the way and see things like, galaxies, star clusters, and eventually the observable universe, which I think is quite extreme to be contained within within a single web page. But here we are. And then we get to the supercluster, and then this is the observable universe. And it's always really fun to just sort of zoom in all the way again and see how long it takes to get all the way down to the scale of atoms. But you can kind of just, briefly fit the whole universe into a single web page, which is what I like about this resource.
Yeah, as you say, useful for lots of things as well as microbiology.
Yeah, exactly. And keeps going. So that's the scale of the universe. It goes all the way down to quarks and so on. And I hope that is useful to many people out there. My next resource has actually, it's much more focused on microbes, and it's produced by two people you might have heard of, or at least a couple people might have heard of them. It's produced by, Dr. Anne Madden, who's a microbiologist and communicator, and a multidisciplinary artist, Amanda Fingbody Pakia. And we can open up this webpage. And it's one of these beautiful designed interactive websites, which basically have wonderful art and images and allow us to explore interesting topics within microbes. So do you have a choice, Jo? Which one of these do you think is the one you want to start with?
Human mouth.
Human mouth? Okay. So, we have, this is a wonderful introduction to perhaps students or kids in a class, and lots of poetic text explaining the kind of world that these bacteria and microbes find themselves in. So in the undulating valleys of our tongue and the deep sea crevices of our gums, the mouth is filled with unique habitats to support hundreds of species of microorganisms. With a constant influx of readily available food from everything we eat, the microbes have access to all sorts of food. Like an open plot of soil that quickly goes from an open field to a field with weeds to a forest of growing trees, teeth of pioneer microbes that land on them, grow, and offer a platform for hundreds of layers of microbial cells to grow on one another. But we have, plenty of different pieces of information and some highlighted species here that live inside your mouth. So, Valonia species. I actually haven't heard of these before.
Violinella.
Violinella, there we go. Learning something new every day.
I did my PhD on oral microbiology, and I did research on it for quite a while after that. So it was really nice to see this.
What are they like in the lab? Are they tricky, or any particular traits or quirks?
I think it's further down. My particular special microorganism was Streptococcus mutans, which have just gone past. So
this one, here we go.
That's the one I did my PhD on. That's the one that was associated with tooth decay.
So they live on the teeth. Okay, so they're very much dental in there.
And I really like the language of this, and it's nice to see the art, but particularly within each section, they provide links to additional resources as well, which are in themselves very informative too. So, it's like the bobtail squid one. You can get links to Ed Yong talking about about microorganisms as well, the diversity of microorganisms. So. There's really good in future activities, citizen science projects and all sorts. So it's great, yeah.
Quite a rabbit hole. You can start getting involved in, you could come here and then end up in many different science
there's
projects.
More that you can do. So it's giving you information, starting off, something that's very artistic, and then and it's writing it in a communicable language, and then it's going into more scientific data. So it's providing really good different kind of levels of information about the communities of microorganisms, and I love that as well, because most microorganisms do live in communities. They don't live on their own on a petri dish. They live in different habitats with lots of other microorganisms. So it's a really nice, it's a really nice way of looking at it. And she's also got sections like in the home and in the shower. And
yeah, let's have a look at the shower.
Yeah.
I don't really think about microbes in the shower, but maybe I will now.
Yeah.
So yeah, I mean, it's a moist environment, isn't it? So
it is.
Plenty of
have
things.
a look at your grout next time you have a shower and see what colour that is.
Well, it's black mold, I can tell.
Well, there you go.
So does it indicate which is the black mold?
Might be.
Okay, let's have a look at these ones.
So
we also had
a lot of slimy ones too.
Yeah. Oh, this is okay. So Methylobacter species produce that pink pigment. That is something I do notice. If you haven't given your sink a sufficient clean in a good amount of time, you do get this kind of pink film.
You do.
Starts to develop. So that's what. Okay. And they produce carotenoids, which is a pigment that you find in many places in nature, not just in carrots, but in bacteria.
Make flamingos pink.
Yeah. Okay. So this is really interesting in the sense that it shows us how microbes live in all our normal world.
Yeah.
And then you can get involved in more projects here. Amazing. Okay. The one I'm really interested in is plant roots, because I live in a country that's plant obsessed. So in the Netherlands, it's very, very much a thing that you have to have your cool plants in your house, right? Everywhere. Yeah. So if I can understand how plant roots associate with microbes, this might give me an advantage in growing my plants, perhaps. But one of the things I didn't think about until I started working at FEMS was the kind of microbiome of microbes that live amongst plant roots, symbiotically with plants, and help them to grow and live. Okay, fun. So, I mean, there's endless amounts of stuff people can get lost in here. Yes. So we'll let them just, explore to their own extent, but it's communityofmicrobes.com. And then, like I say, all these resources are available on the opportunities board of the FEMS website. So you can find them all there. Right, the next one is another kind of similar, well, not that similar, but it's another beautiful interactive website which you can explore. It's called Bacterial World, and it comes from the Oxford Natural History Museum. And it's got two sections. We'll just show you the normal website to begin with. But it's all based on an exhibition that was happening at the end of 2018, beginning of 2019. And it too basically shows you the untold story of bacteria, and just has some of the most beautiful images. I mean, this is a petri dish with some kind of bacteria or mold perhaps growing on it, and the colors there are amazing. And again, it's another place that you can perhaps set your class on or ask people to start some research from, and it just kind of outlines in a visually impressive way so much information about the microbial world. So one of the things it likes to point out is that if you are sort of counting species amongst all life forms, 78% of them are bacteria. And all the plants that we've ever known are 0.02%. All the animals, 7.3%, fungi, 7.4%. Protists, this kind of, I don't know, oddball grouping of microbes and animal-like things is 7.3%. But yeah, 78% of these are bacteria. Now, I wonder if they have included archaea in bacteria, because I don't see archaea in this.
Oh, it's on that little table on the graph there, isn't it? So
Yeah, okay. No, they
Yeah,
are keeping it separate.
yeah,
So yeah, bacteria really are very numerous.
yeah, and
And
yeah.
one of the oldest branches. So they branched off well before we split off from archaea.
Mm.
I always find that quite bizarre.
Yeah. It is an absolutely gorgeous website, and also you've got the links at the top, so you can go straight into different topics from the top rather than sort of racking down all the way through. And there is another scale, a scale zoom in there as well. So there's a press start there. See what? Yeah.
Okay. Bacteria Explorer, dive into the bacterial world. So, wow, we're going to learn about all the different forms, because actually bacteria do have lots of different shapes.
Yeah.
So
okay. It's a similar
like thing. Yeah, cool. Okay.
But it's obviously focusing more on the microorganisms and the scale of everything. So,
I mean, this is all really great because you might not have a microscope in your house. It's not as common.
No.
So, getting to grips with scale and size is something that is hard to do without a microscope, but much easier now you have these kind of tools.
Yes, absolutely. Yeah.
So let's have a look. When do bacteria start to pop up, or are we reaching the end? So here we go. Select a bacterium. Right. Do you have a favorite one that we should start with, Jo?
Oh, look at that one. Sorry, the spidery one. Just
this one.
Yeah.
This one in the middle?
Okay, yeah, whichever really. They're all.
Which is the spidery one? Do you mean this one?
The blue, yeah.
Okay, yeah. Branched.
Yeah, so they're looking at the different morphologies then, aren't they? So and helping you to sort of classify and find out more about the different types of microorganisms. So these ones are particularly common in the soil, and they produce a lot of our antibiotics, streptomycin. So you can find out, so the other one gives you the overall scale, and this perhaps gives you more about the diversity of the microorganisms, of bacteria particularly.
Yeah.
So, yeah.
But it is a bacterium, so it's not a fungus. It's not anything that you would expect
because it's a eukaryote. Yeah.
That's another new thing I've learned today. I didn't really think about the possibility of branched bacteria. I always think
they are pretty good, pretty versatile, really. Yeah.
Yeah.
I think you've got one of them on the 52 organisms as well, actually.
Oh, maybe we do.
I think so. Yeah.
Hard to remember them all. Look at a spiral one, because that's always pretty fun. Spirochetes. So these are usually, swimming, and the spiral, much like a corkscrew, can help a ship move along. The same shape works for bacteria. Right. Human immune system. Okay. So this is a tricky thing. In spirochetes flagella, which normally are very obvious to our human immune system, are internalized, making them a bit harder to detect. Yeah, okay. I mean, there's just so many wonderful things on this website, and we're just at one end of one. Let's have a look at one more quickly. How long have we got? Okay, we've got a few minutes more that we can play about here. But again, we encourage anyone to spend time on here, or to share it with people who need to learn about microbes or want to learn about microbes. So I guess the sphere coccus shape is a very classical bacteria shape. Tiny spheres drifting around. So yeah, they're not usually able to move, so they just kind of float around.
Not really green and shiny either, I don't think. But...
No, probably not. I mean, maybe there are some cyanobacteria which perhaps are a bit.
Yeah. And there's the bottom one, Prochlorococcus. So those are probably.
Ah, there we go. Yeah. Okay. So this one is. There we go.
Yeah. So they might be.
Very nice.
Yeah. Okay. Yeah.
So actually, this brings us on. This is a sort of another section of this website. But I originally learned about this section when I was, back in the world when we could travel. I was at the Danish Microbiology Society, their national congress, I should say. And one of the keynote speaker, whose name I'm really sorry I've forgotten, who has some amazing research, showed us this gut wars game. So this is a fantastic way to show people how bacteria and microbes in your gut are living and competing for the finite resources there. So in this case, we just take, we're going to have to go against Speedy, which is a fast, its strategy is to divide fast, to reproduce quickly, let's try and just reproduce faster than its rivals.
Yeah.
To defeat Speedy, we have the choice of 4 different bacterial strategies. We can be slimy, we can be stabby, we can be toxic, or we can be vengeful. Do you have a particular?
I do love a bit of slime myself.
Slime. Okay, let's see if we can slime down Speedy and take over the gut microbes. So this is what happens all the time. If you eat a piece of food that contains microbes that you haven't had in your gut before, there's a chance that they could outcompete the native microbes. I mean, the ones in your gut might have a better start and a bit of a foothold, but it's entirely possible that the composition can change. So let's simulate the battle. So we're slimy as these yellow ones, Speedy as these blue ones.
This is what I love, the way that you've got this biofilm development. So it's a really nice interactive and changeable image of a biofilm, looking at the microcolonies and the gaps between as well, and how different organisms grow. So that in itself is wonderful. Yeah,
and it shows you that, you know, there might be kind of, you know, areas of more dead microbes as well and more live microbes as the biofilm grows. So here we go. The winner was slimy. So
well, there you go. And a bit of glue never goes amiss, does it?
Doesn't, does it? Let's do one more pairing just to see if slimy wins all the time. So we need to defeat slimy now. So obviously not Speedy. We know that's not going to be a good choice. Right. Stabby, toxic, vengeful. I'm going to pick toxic. Maybe it can get through the slime and kill off slimy. So you can see the little bits of slime are these kind of grey blobs, but maybe it'll protect slimy from our toxicity. Oh no, the toxins have built up and they've killed off all the slimy bacteria.
Yeah.
So in this case, toxic was a good strategy, and it's pretty much taken over the gut. So this can happen when you get ill or feel ill. New bacteria can enter into your gut, kill off helpful commensal bacteria or microbes.
Yeah, you won then. There you go.
Well done, toxic.
I think, you know, games are a great sort of opportunity to get people interacting and thinking about science. So we developed one called Simfection as well, which is looking at communities and how vaccination can work if you change different parameters. But also, I think there's a website called Game Doctor, and they've produced a lot of different games as well with microorganisms. So there is quite a market, and there's also board games as well that people can use to play for microbiology, like Pandemic, and there's plenty of others as well. And Top Trumps games, which I think FEMS have produced previously.
Yeah, there are some.
A set of cards, haven't you?
Set of cards. I've got in my flat. They're not in this room. That's a shame. Missed opportunity. But I do remember playing Pandemic. Well, I think there's an online version called Pandemic 2 several years ago. And the role is to try and, seems a bit macabre now, but the role is to try and infect the world with a new pandemic as fast as possible and as extensively as possible. You can choose different microbes or different pathogens, from parasites to viruses to bacteria. I think you can also pick fungus, but they all have, you know, different restraints on how fast they can spread, how damaging they can be, and how easy they are to treat. So you have to kind of pick and choose between these different playoffs. But yeah, games are a great way. And so I encourage anyone out there who is a microbiologist who has the ability to code to produce as many fun games as they can.
And I think another thing that's important is, if you are a microbiologist and you have tried different things, write it up as a paper as well. You know, we've got journals. So there's FEMS Microbial Letters, the professional development section of it that will publish different activities, and then you can show something that you've developed and how you've evaluated it, and then get that published, and then that will help other people and provide other people with ideas. I think there's an education webinar going on, isn't there, at 10 o'clock as well? Is that from...
it's 10 o'clock US time. I think it's 3 o'clock UK time and 4 CET, Central.
So, you know, there's more examples of using social media in education in that as well, and Twitter and others. So, you know, that there is, as I said right at the beginning, the ingenuity of microbiologists, because that's who we're focusing on today, is staggering, really. And we just really need to, as much as possible, tell other people what we do, other microbiologists and educators. It helps everybody, I think.
In fact, there's a guest I've got later today, similar, I think it's the same time as the Twitter webinar. So if you want to learn about Twitter, go to the webinar. This is an Instagrammer. So if you want to learn about Instagram, come to the Microscope Hour. And yeah, so he's actually published in FEMS Microbiology Letters a paper about his Instagram experience. So it just gives you a bit of a brief introduction to what worked and what kind of videos have been really popular. And by popular, we do mean big numbers. I think he's nearly got 140,000 followers just following him for videos of microbes. So there are amazing methods, whether it's games or social media or video, for getting huge numbers of people interested and excited by microorganisms. Because it's not something that, unless you have very specialist access to expensive equipment, that is very easy for people to experience. I mean, we're quite lucky that we get, we've had experiences with light microscopy and electron microscopy and fluorescent microscopy, and the idea of looking at these things close up and in detail just seems quite normal, but it's actually an incredibly rare experience. So there are ways to get that out to as many people as possible. And yeah, we encourage you to do it if you can.
Absolutely. And another way is to use the word zombie. That usually helps. If you kind of talk about zombies and use them as a model for infectious disease, that kind of brings me back to my literature book club and young adult novels. You can use the zombie as a model for infection, and then you can criticize or comment on the effectivity and compare it with real microorganisms. So we've had quite a few, we wrote a paper about the modelling of a zombie apocalypse once, and that actually got quite a lot of readings as well. So that was a simulation called Sim Zombie.
What were your conclusions? Should we be scared and fearful of zombies?
It depends how fast they are, I think. So if you look at the old literature, they were pretty slow, but they get quite fast. Once you got 28 days later and beyond, they got a lot faster. But they're quite boring in the end, because they're so kind of vicious, really, I think. So it's better to have something that you have a chance of surviving, obviously. Yeah.
Absolutely. There's one more resource I want to cover before we have some other guests join us who are authors of books about microbes for kids, so very much on topic. But this is a project by the Croatian Society of Microbiology, one of our member societies, and it's Microsium, which is a virtual microbe museum, and they are actually giving us an hour presentation later today about the project, what they learned, how they did this, perhaps some experience there for people who want to do something similar. It's at noon till 1 UK time, and they have 3 different talks by people involved in the project. But and this isn't the only output, one of the outputs of the project was this Microsium museum, which tries to show people, how microbes in their own house and in their own home are incorporated into their lives. And it's available in Croatian and English. Obviously, this is the English version. But let's have a look down in the living room. And what we can do is we can look over different sections and learn a bit more about what's going on here. So here we're learning about how, we are full of microbes. We are our own microbes. And it provides lots of interesting information and links to new sources and, fantastic facts. If we could collect all the germs and the genes from our gut and put them on the scale, they would weigh about 1.5 to 2 pounds, or a kilogram if you're in the metric system. Which is kind of astounding if you just think about, macroscopic masses of tiny microscopic things.
Yes, it's a really lovely website, and it's attractive for younger viewers as well, I think. And that's another interesting issue. It's who your audience is and how you can modify different activities to the age or the experience of your audiences. And many of these websites adults are very interested in much of this, as well as children and young adults. So the versatility of lots of these activities is significant as well.
And it's also a great opportunity for artists and scientists to collaborate, because you have to get people who are creatively talented to produce beautiful illustrations. You have to get people who are, epistemologically or empirically talented to get the knowledge, and combining them together produces something which is better than the sum of its parts. So
absolutely, to help communicate,
encourage more of this if possible.
And you've got your microbe art competition, haven't you, as well?
Actually, let's just turn to that for a second before we have our next guest on the line, because I think some of the winners have been announced.
All right.
So in the past few hours, we have learned that the winner of the Agar art competition is Naomi Matthew with this fantastic
Oh, wow, yeah.
Peruvian man, which is, I think just blew everyone out of the water.
Yeah.
So congratulations to Naomi. And the best cartoon from Joanna Carvalho, who you can also find on Twitter, is this wonderful, wonderful illustration of different kinds of microbes living in bubbles. And we can already now understand a few more details about the shapes. We've got some spirillum, we've got some flagella, we've got the spherical coccus-shaped bacteria, and yeah, beautiful illustration here.
Yes, lovely.
And just announced 9 minutes ago is this wonderful piece by Sarah Adkins Jablonski, which is made from kind of superimposed images from agar art, put into this beautiful kind of illustration image here. Yeah. So well done. All of the microbe art winners will be receiving prizes, microbe themed prizes. We'll announce those after the day is done, and we can contact all of the prize winners and just make sure we have all their details. But they will all be receiving some prizes, and details on that coming soon. So well done to everyone who entered, and well done to the winners. There's more to come. I think they're every half hour between now and 1 o'clock.
Wow, great.
Right, we have a couple of guests who I shall just bring into the call, who are some young authors of books. Let's just stop sharing, about microbes. So let's just make sure we've got everybody here. So Sarah and Katie.
Good morning.
Morning. So you'll start muted. So if you can unmute your microphone, just to check everybody's here.
There we go. Can you hear me?
Yes.
Yeah. Yes.
Absolutely. So we've got Sarah Hull, who is the author of See Inside Germs, and Katie Daynes, who is the author of a younger title, or for younger kids, I'm guessing, or is it just a younger title?
For younger kids, definitely.
Younger kids, yeah, called What Are Germs? So I'm just going to hand over to you guys to give a brief introduction about each of these books, and then I have some questions for you, as always. But if you share your screen, you should be able to give us a little presentation and about these beautiful books.
Okay, great. I'll share my screen now. Just want to say it's such a treat to be invited along, and that Sarah and myself feel a bit of imposters here because we don't have a background in science, let alone in microbes, but we do have expertise in children's books. So we
were just saying that how important it is that people need to work together actually to communicate. So that's perfect.
Wonderful. Yeah, so we hope we can take a complex topic, and with expert input, we can then turn it into something that's accessible for children. So I hope you can see my screen. This is a summary of the books we've got out already and in the pipeline. So See Inside Germs, Sarah will be talking about. That's more kind of key stage 2, so age 7+. What Are Germs? That's a young 3+ preschool book. And then What Is a Virus will be coming out next year. So more reception, key stage 1. But we'd like to say with all our books that you're never too young or too old to read and find out more from them. So I'll just pass over to Sarah, who just need to unmute yourself, Sarah.
Great. So I'm talking about See Inside Germs, which came out this year, but I actually started working on it more than 2 years ago, so long before anyone had heard of COVID-19. It's 16 pages with lots of flaps to lift, more than 100 in fact, and covers a lot of ground, everything from what germs are and microbes to how germs spread, vaccines, antibodies, antibiotics, and even antimicrobial resistance. If we could have the next slide, we can have a bit of a look inside. So a big part of what we do to make these books is about combining information with illustration to make them really appealing and accessible to our audience. And this is especially important for a topic like germs, where there's lots of tricky concepts that our readers won't have encountered before. And of course, an added complication is that a lot of the things you're talking about are far too small to see. So writers like us will work very closely with in-house designers to develop layouts for the spreads, and it usually takes a few attempts. And so I started this book once I'd done a bit of initial research by coming up with concepts for the page with the designers. So we decided the virus page would be set in a factory, and the spreading germs page we wanted to set in the real world to show how things, how germs can really spread around. And here on the page you can see we decided to talk about good and bad bacteria, so that I wasn't just giving a negative picture of germs, but also showing how bacteria and could do useful things for us. On this book we worked with the illustrator Peter Allen, who as you can see he's added a lot of energy to the pages with his spiky drawings, and he was really brilliant at giving the microbes lots of character. And you can see we've got little bacteria wearing chef's hats, and they're the ones involved in helping make food, and below them there are bacteria cleaning up sewage and oil spills with lots of cleaning equipment. And another thing I wanted to mention about this topic was that we needed to try and strike a balance between talking about scary diseases and making sure we weren't going to give our readers nightmares. And sometimes this could involve a bit of to and fro. So for our illustrator illustrated a flap about plague with lots of dead bodies on, and we had to remind him about the audience and get him to take them out. The flaps are obviously really important to how See Inside Germs works for readers. They make the pages really dynamic and allow readers to explore the material as they choose to. Flaps can reveal surprising facts, or a punchline tells stories in a comic strip form, and they're also really useful for breaking down complex ideas and making them quite simple. So the example up on the presentation at the moment is about how bacteria turn into superbugs, and here we just used 3 flaps to explain how these mutations happen. And obviously Peter's funny illustrations which show the bacteria randomly gaining a shield and then developing into a big army of armoured bacteria. So if we move on to the next spread, this is the spread that little snippet comes from, which is the final spread of the book. And obviously superbugs are really quite scary, and I didn't want to end on a scary note, to come back to my point about not wanting to give our readers nightmares. So I decided to end off with a mention of microbe heroes, some of the microbes that might help us fight superbugs and also tackle other problems like plastic pollution. So they're quite fun. And I also wanted to end with a call out to our readers, suggesting that they could become scientists and find new antibiotics to help us tackle these problems.
Thank you very much, that's good. More scientists, please.
Yeah, I'm very, very happy to see you showing the good side of microbes as well, because, you know, especially this year, there's a lot of bad press around microbes.
Yes, yeah.
But showing all the wonderful things they do is, you know, a fantastic thing. In fact, in the live stream that's already happened, we had talks about bacteria degrading plastics and cleaning up oil spills. So it's really nice to see those in the book as well.
Yeah.
Is there a link to, does Usborne provide resources that support these books as well, did I see?
There are, I think, lots of our books are internet linked, so there'll be safe places on the internet where children can read a bit more about things that come up in the book.
Great, that's great. Thank you.
So yeah, I had a couple of questions. So you mentioned that you you try and work with experts to try and get these books as accurate as possible. So how do you find that experience then as a non-expert? What's the most challenging and the best thing about working with sort of scientists?
Well, I think it's very reassuring to know that the things that I've decided are interesting will be read and approved by someone who knows their stuff. And it can also be, we like to get them involved quite early on as well, so that they sometimes mix things up early on. So my expert, who's a clinical medic, suggested that we might need to explain what a cell is right at the beginning of the book in order to tie everything together, and that was really useful. Yeah.
Cool. Another thing is, I mean, so you also have lots of artists produce work for you. So do you have to connect the artist and the scientist up at any point during the process, or do you act as like a go-between?
We usually act as a go-between, but the expert will look at all the final artwork just to check that we haven't made a bacteria the wrong shape, or that our phagocytes have the right shape nucleus, or details like that. They'll make sure that all of that's correct, but then we feed that detail back to the illustrator.
We always use photo references where we can, and if it's something really obscure that we struggle to find, then we'll ask the expert, can you get me a reference for this particular microbe or situation. So it's quite an important relationship, that one, with the expert, that we feel it's someone we can bounce ideas off, and also that they take us seriously, even though it's for a really young age, that we're really passionate about getting good science across, but sometimes we do have to give them smiley faces and make it approachable. So yeah, it's a balance there.
I like the smiley faces on the bacteria. I know it's not strictly correct, but it is fun to to personify these things in a way.
I'd like to see a few of the younger book, just whiz through.
Yeah, let's have a look at it.
Yeah, so just to give an example of taking a topic really, really young, and again, how important the illustrations are, and the text alongside, and how brilliant flaps are at allowing us to go inside things, zoom in on things, animate the scene. We've got there are more germs than you think in the kitchen sink here, and then you open this massive flap and the germs are multiplying and having a party in the sink. And here we've got how can you fight germs, so looking at natural defences, getting the flaps to open so you've got inside the nostril, and all those kind of yucky bits that the kids like the bottom right there, the germs being killed off by stomach acid, but done in a palatable, amusing way, hopefully. And I didn't know whether to include this actually with such a professional audience here, but I just wanted to show how we tried to boil down even explaining how a vaccine works. So making the syringe into the teacher who's teaching a class of white blood cells how to make the right antibodies to kill off germs. So yeah, hopefully we managed to get enough of the proper facts and condense them down so that they're approachable, accessible. And then this book is the one that's in the pipeline, so coming out next year, but that's been my lockdown really, is trying to get out a book on viruses, which a year ago we'd never dreamed of doing at all, probably, let alone for this really young age group. So it's a mark of how the world has changed.
Yeah, really important. It's great.
What stage then is this book in the pipeline? So you've begun drafting the text. Are you working with the artist yet, or I don't really know how the pipeline works? It's finished. Okay.
And it's gone. I haven't seen the proofs yet, but it's been approved by the expert, by everyone in-house. And what's quite remarkable for this title, and again a sign of the times, is that 15 other languages want to jump on our first print run. So normally they'll wait to see the book, and whether they like it, whether their markets will want it. But this time they're just going for it. And that's been the case actually with all our microbe books in the last year, just different languages coming on board, wanting to print it because of the huge importance, especially the hand hygiene with younger children. So there's just an example here of how recently South Korea have printed the Suicide Germs book, and everybody who buys it gets a free face mask strap, because that's a big thing in South Korea apparently, accessory. But yeah, that's it from our presentation, so I'll stop sharing.
Awesome. Thanks a lot for joining us.
Can we just ask the virus book, you said it was coming out in 2021. Is it going to be available for the Christmas market or not then?
No, unfortunately, for this year, no. It will be, I think March is the scheduled publishing date, but if we can whiz it through quicker. It's a long-winded process, the
Yes.
printing side, but certain printers can work faster, so I think they might try and get it out earlier if possible.
Yes, I think so. Thank you. That's great. Thank you.
Awesome. Right, thanks for joining us. We've got 5 minutes before our next session, so I was just going to run through the education resources section on the International Microorganism Day website, because we have done, and you don't have to stay for this, Katie and Sarah, but
Okay.
you can leave at the moment. But thank you so much for joining us. And
Thank you for having us.
find this book, I guess, these books in all good bookshops, which is something I've always wanted to say. But there's one final thing that we can show before we move on to our next speaker. And this has been, yeah, I guess a new thing for International Microorganism Day. In a very similar way, we've kind of combined artists and scientists and researchers, and we've produced 2 educational resources for IMD, and they're here and they're free, and you can download them whenever you want. And they are just here in the education resources section. Hopefully in time we can expand it, but at the moment we have one on the Winogradsky column, and one I mentioned earlier with our wonderful microbiologist Michael Sauer is how to make sourdough bread. So I'll dive into Winogradsky, who was a Russian microbiologist who kind of paved the way for thinking about environmental and ecological microbiology. So one of his experiments was to build these kind of columns. So you can use lots of just general household materials you can see here. You have to basically mix together a kind of strange substrate here with eggs and paper and mud. And you build this kind of layered column within a plastic bottle. You expose it to things like warmth and a little bit of sunlight. And yeah, the idea is you take pictures of it every week for 8 weeks, and you set up your mixture with different combinations. And basically what happens, and it's a shame to give away the ending, but you develop a layered structure of bacteria living in a community that all have vibrantly different colors. So you get to see this wonderful sort of demonstration of how microbes organize themselves into different communities within an environment. And all of these resources have the main infographic, which is useful for teaching, but then they also contain separate teacher and student instructions. So if I just load this up, we can have a look. So this is what you would give to your class. It seems to not be working right now. Oh, here we go.
Are they, is there an opportunity for people to post their photographs on a website anywhere?
So you can always tag us at hashtag International Microorganism Day, and we will retweet you. If you're on Twitter, or if it's on Instagram, we can put you in our story, or if it's Facebook, we can share you as well. Yeah, so if you want to do any of these experiments and have a bit of interaction with us, just simply use the International Microorganism Day hashtag, and we will retweet your pictures. But yeah, this is what you'd give to the class, some nice instructions, and then there's a special teacher's pack with a few bits of material for the teacher, and also some kind of health and safety stuff about how to make sure it's sterile and how you're not going to kill anyone. So yeah, hopefully people can make use of this. The other one I went through a little bit earlier was how to make sourdough, very similar kind of design and infographic, making the culture, feeding it, keeping it going for a few weeks, and then using it to eventually make bread. And there's also a little bumper pack here for another kind of thing you can do to show how sugar leads to carbon dioxide production in yeast. And again, there's a teacher and student pack at the end if you want to do this with a class. So yeah, both available here. 2 books available in the shops. Hopefully there's lots of materials on the website that we've run through over this past hour to help you teach and explore microbes to a whole range of kids and students. And
also a great opportunity to share if you're doing anything anything yourselves as well. And FEMS are here to kind of help to disseminate, and FEMS member societies also produce lots of things too. So it's microbiologists are very, very dedicated when it comes to education and communication, I think.
Yeah, absolutely. And it's so important as well. It's one of these things that really does make a big difference in terms of public health and, you know, lifestyle.
And science literacy generally as well, I think. Yeah.
Exactly.
Yeah. Yes.
So yeah, check these out if you're a teacher or if you're just interested. Our next speaker has arrived in the waiting room. So Sarah and Katie and Jo, I must say goodbye to all of you.
Lovely to see you.
Thanks for joining me. Okay. And yeah, enjoy International Microorganism Day.
Thank you very much. Enjoy the rest of your day.
I will do. Enjoy yours.
Bye.